How's it going, everyone? It's Chow here, and today we are going to talk about light-independent reactions. So let's get started. Okay, so we talked about light-dependent reactions in another lecture, but this time we're going to talk a little bit more about the light-independent reactions. So light-independent reactions also occur within the chloroplast, which is where photosynthesis takes place, but instead of being on the thylakoids, the light-independent reaction, which is also known as the Calvin cycle, is going to occur in the stroma, which is over here. The overall byproduct, so the overall product of the light-independent cycle is sugars, carbohydrates. And then they're often going to be 3-carbon sugars, which can be eventually used to make, for instance, glucose. So when we look at light-independent cycles, there are actually some scientists whose names are Calvin and Benson. They used 14-carbon radioisotopes to basically determine the sequence of reactions in that whole process of CO2 fixation. So what they did was they exposed chlorella to, which is an organism, to 14 CO2 and then extracted organic compounds and separated them by paper chromatography. What they found out was that the results only showed the 3-PG or 3-phosphoglycerate, which is a 3-carbon sugar phosphate, on the results and the really initial results. So what it showed is that only 3-PG was produced at the very beginning. And this suggests that the first product of CO2 fixation, the first compound to be formed in the Calvin cycle, is 3-phosphoglycerate, which is again a 3-carbon sugar phosphate molecule. What's really fascinating about 3-phosphoglycerate is that it's also an intermediate in glycolysis. Okay, so the pathway of CO2 fixation. So what really happens is that you have a CO2 molecule, so a carbon dioxide, that's added to an acceptor molecule. So this is the beginning of the Calvin cycle. So the carbon dioxide, through a carboxylation reaction, basically combines with a ribulose 1,5-bisphosphate, also known as RUBP. So carbon dioxide combines with RUBP to make this 6-carbon skeleton over here, through the mediation of an enzyme known as Rubisco. Once the Rubisco catalyzes this reaction, we actually see something happen, and that is this 6-carbon intermediate basically immediately breaks down into 2 3-carbon molecules. And these 2 3-carbon molecules are called 3-PG, or 3-phosphoglycerate. So we had this thing that we talked about where we produce 3-PG. This 3-PG, this 3-phosphoglycerate, is what happens over here. So again, first steps of the Calvin cycle is you take carbon dioxide and then you add that carbon dioxide through a carboxylation reaction to ribulose 1,5-bisphosphate. And then once it's added through the mediation catalyzation by Rubisco, you actually form this 6-carbon intermediate that immediately breaks down into 2 3-PG, or 3-phosphoglycerate. So 2 3-carbon molecules called 3-phosphoglycerate, and you see that phosphate group on there for obvious reasons. Yeah, that's correct. Yeah. Okay, so what's really interesting about Rubisco is supposedly it's the most abundant enzyme in the world. That's a trivial point that supposedly is needed to be shared. Now, the Calvin cycle is going to be consisting of 3 different processes. So the first process is basically fixation. There's 3 steps that we have to go through. The first step is fixation of CO2. The second step is reduction of 3-PG to G3P, or 3-phosphoglycerate, into glyceraldehyde 3-phosphate. We then have to regenerate that RUBP at the very beginning. So in these 3 processes, we see some things that are very interesting. So Rubisco's over here, and it does its thing, makes those 3-PG molecules. And then in step 2, we see a reduction reaction where we turn an acid to an aldehyde. So it's a reduction reaction, and we use 12 ATP and 12 NADPH. So these ATP and NADPH molecules, so these 12 ATP and 12 NADPH molecules are ones that were made in the light reactions or the light-dependent reactions that we discussed earlier. And so we use those ATP and NADPH up, and we make a reduction reaction to turn a 3PG into a G3P. And then in step 3, we're basically using 6 ATP to reform the 6 to reform RUBP. So we're taking these molecules over here of G3P, and we're going to use those G3P molecules and reform RUBP, or ribulose 1,5-bisphosphate, which is the starting initial acceptor molecule of the Calvin cycle. Hence, we reform the cycle. So one thing to think about is that the Krebs cycle, we remake oxaloacetate at the very end. Same thing happens here in the Calvin cycle, where we remake RUBP at the very end, hence the reason why it is a cycle. So what's really fascinating is that it's steps 2 and 3 that are going to be using energy in the Calvin cycle. Ultimately, the product of the Calvin cycle is glyceraldehyde 3-phosphate, which is shown over here. And these G3P molecules can be used to make glucose and other carbon compounds. Now, if we look at the Calvin cycle as a whole, and maybe just the entirety of the light-dependent and light-independent reactions, what we see is that there's going to be problems with efficiency. So, there are energy losses that occur during photosynthesis. So if you have 100% of the sunlight coming in, only about 50% is going to be absorbed by photosynthetic pigments. And then only 30% is going to be absorbed due to plant structure, and so on and so forth. So the efficiency of photosynthesis, while it's pretty good, isn't actually that great if you look at the statistics. So there's going to be parts of that energy that are not going to be used to their max. But that's trivial information. This is something here that can be useful that I'll just end and summarize this entire lecture with, and this is basically all these different pathways that are connected. So you can see how the CO2 molecules in the Calvin cycle, which we turn into 3PG, and then of course G3P, how these molecules, for instance 3PG over here, we'll talk about 3PG first, can be used to make glycerol, it can be used to make pyruvate, and then this glyceraldehyde 3-phosphate product of the Calvin cycle can be put into these processes of glycolysis or gluconeogenesis, and so on and so forth. So this is just a good summary of how all these different cycles and processes are connected. So that's basically it for the Calvin cycle. It's nothing too difficult. It's mostly this slide here that you have to keep in mind perhaps. So you start off again with the fixation of CO2, you reduce the 3PG to G3P, and then you regenerate RUBP. And then, of course, the result, the main key product here, is a sugar called glyceraldehyde 3-phosphate, also known as G3P. So there you go, the light-independent cycles, or also known as the light-independent reactions, also known as the Calvin cycle. There we go. So I hope you all found this useful, and as always, best of luck studying, and I will see you all in the next video.